Okay, so these series of slides look at the prevention and treatment in parasitic diseases. So looking at prevention, there's three layers or steps of prevention. There's primary prevention, secondary, and tertiary prevention. So primary prevention aims to preventing healthy people from becoming sick. And so the way this is done is by promoting health, preventing exposure, and preventing disease, whether that's being vectors or the organism in their environment. Secondary prevention is aimed at early detection of the disease, so testing populations so that you can identify early on before symptoms even start whether the organism is present in that individual, and then you could start treatment. Also, you could take steps as far as isolating them. That leads back to primary prevention. With tertiary prevention, this is the treatment of someone who has the disease. So again, isolating them so that we don't get healthy individuals sick. Also, making sure they don't return to the population until they are no longer infectious. And so you can see how these different steps actually work together. And you can also see how, even though COVID is a viral disease, how we can see some of these strategies, these prevention methods are in place by some of the public health guidelines for COVID. So an example of a parasitic disease would be the Guinea worm. And so the Guinea worm has reached eradication, so it received a certificate of eradication from the World Health Organization. To get their certificate, you have to have 3 years of no cases. You get a pre-certificate and then additional years to get the full certificate. And so Jimmy Carter was a key player in working in getting the eradication of Guinea worm. And so there's this link for the NPR interview. You also have access to the transcript on the Canvas page files. So I highly recommend either reading or listening to it. It is fair game for the exam. So, and it just, it's again an interview with Jimmy Carter talking about his involvement through the Jimmy Carter Center with the eradication of Guinea worm. So I've had the pleasure of going to the Jimmy Carter Presidential Library, which is also connected to the Jimmy Carter Center. They're on the same campus, and a large piece of what Jimmy Carter did was looking at public health diseases, not just ones in the U.S., but globally, and working in making sure that they were eradicated or under control, because he was aware that when people's health is in jeopardy, that can affect a lot of other aspects of their quality of life. So in class, we looked at this review article looking at the guinea worm and its eradication. So guinea worm's unique because there's no drug or vaccine for it. And so strategies in eradicating it are slightly different. So obviously if you have a drug for treatment or you have a vaccine, you can use different approaches. So if you have a vaccine, you can just make sure that you have over 70% of the population vaccinated to certain herd immunity. If you have a drug to treat it, then you can make sure you have access to that drug, so when people become sick, you can then treat them. So with guinea worm, neither of those are available. So the strategy, if you have a guinea worm infection, is to remove it. And so the way they do that is they coax the worm out, and then as it's exiting, they wrap it around a small twig and continue to pull it out. The problem is because of the life cycle of the worm, it can release eggs into water, and so if people are wading into the water, they can become infected with the worms. And so if we look here from this figure from the review article, we'll notice that there's a number of different steps that can be used to stop that life cycle. So, case containment, if you know someone who has it, making sure that they're not going into any of those waterways so that the parasite can't be released into the water. Vector control. So the cyclops is the vector for the larvae, so it actually eats the larvae, and then so if you treat the water so that the cyclops aren't present, that acts as a vector control, can stop that part of the life cycle. Making sure that people have access to improved water sources. So we see this person collecting, it's going to have the cyclops in it, so then this just, if they drink that, then they're going to ingest the larvae. It becomes an adult, and the cycle completes. So making sure that they have clean water sources, whether that's putting pumps in, so instead of them having to go to ponds to collect that, they might have a pump for underground, that'd be underground water source, which is being filtered. Also being filtered through the sand. Also just the use of filters, whether these are straw filters or filters placed on top of jars where they're going to collect water, so that they're not drinking the water directly from the source, but there's actually going through a cleaning process. So then the last step would be making sure that you're doing surveillance, so that you're aware of cases that are present, so that instead of getting to the point where a high number of the population are infected with Guinea worm, that you're catching this early on, making sure that you then ramp up other steps, that if you do see an increased number of cases, that you're taking steps to control the vector, you're making sure that people have access to clean filters, maybe a pump has been contaminated, or maybe the pump doesn't work, so now people have gone back to the fresh source. So surveillance allows you to ramp up other pieces. And I guess the other piece that's not on here is making sure you're educating the population. So having usually trying to educate people inside the community to then educate additional people. So a lot of times, outsiders are not going to be received in the same way as someone within inside the community. So if there's a trusted medical professional or caregiver within the community, having them do little seminars about ways to prevent the spread of these for guinea worm, but also for other parasitic diseases. So there's a number of anti-protozoan drugs, and the mechanism of actions vary depending upon the drug. And so some of them may inhibit electron transport to the mitochondria so they can't make ATP. Others will inhibit folic acid synthesis. So that's a common pathway that's used for for anti-protozoan drugs, but also for antibacterial. So one of the reasons for that is because we as humans don't actually produce folic acid, but some of lower order organisms do produce their own folic acid, and so drugs that target that would affect them but not us. Okay, others will work by damaging producing damaging reactive oxygen species. We can also inhibit DNA synthesis, and we can also inhibit heme detoxification. Okay, and so these different mechanisms of actions gives us kind of a repertoire of drugs to use. Not all drugs are going to be effective against all protozoans, so some might work better against other ones. So they would be used depending upon that. Chloroquine, which is a heme detoxifier, is one that's been kind of in the news. So it's an anti-malaria drug. But you can imagine having your heme detoxified. If you're taking high doses of this, would not be a beneficial thing. Metronidazole, which inhibits DNA synthesis, is another one that's a pretty common drug. Your sulfurs, sulfur diazine would be an example of a folic acid. And again, sulfa drugs are used also for bacteria, not just for protozoans. So for our antihemolytic drugs, they are going to work off of different mechanism of actions. So this is where microtubule formation or glucose uptake can be inhibited, reduced. We can also block neurotransmission. This is a popular mechanism. So in these drugs, this actually causes the parasite to become paralyzed, and it is either paralyzed in a way that it can no longer hook onto the intestine, and then it gets passed. But this paralyzation can also lead to starvation for the organism. We can also inhibit ATP production. We can cause calcium influx, and we can inhibit RNA synthesis. So mebendazole is a common one that's used. Again, ivermectin, and we'll talk a little bit more about ivermectin in a moment. So one of the reasons I like to talk about ivermectin a little bit more is because I get to talk to Dr. William Campbell. So Dr. William Campbell was one of the researchers working at Merck that worked on the discovery of ivermectin. So he was awarded the Nobel Peace Prize for his work. He was also a speaker at Darwin Festival in 2017. And so he talked about river blindness and the story behind its prevention, which is the story of discovering ivermectin. I've linked his Nobel Prize lecture there so you can listen to his talk. He gave a very similar talk, a little bit like more updated than when he actually talked about how like receiving the Nobel Peace Prize, and when he spoke at Darwin Festival, he spoke about it being a group effort. So it wasn't his work alone. It was this collaborative project that they worked on together, and that's how science happens, and that it really kind of happened in this unplanned process in some ways. So what they had taken is they had taken soil samples and taken fractions of it, and basically just took those really crude kind of preps and then injected them into mice that were infected with the hemolysis, and they then looked to see which mice recovered, and then they went back to the fractions of the recovered mice, and then they went and said, okay, well, what chemicals are present in here? And that's when they came across ivermectin. And since then, so in 1981, ivermectin was launched. So it is still used in veterinary and human medicine for a number of different hemolytic diseases. So HeartGuard has ivermectin. Also, if you have horses or work around horses, usually ivermectin is one of the dewormers that's used. So at least with horses, a lot of times that they'll rotate the deworming medicine so that you don't build up the resistance. So there's definitely some resistance to ivermectin present. So there's genes that are going to give these parasites a way to either pump out ivermectin so it doesn't have an effect on it, or they have different pathways that circumvent the pathway that ivermectin is going to affect to cause that paralyzing effect. But it still is effective today and used, and it's also used in human medicine too. So hopefully you found this audio lecture useful in understanding the prevention and control and treatment, the controlling of parasitic diseases. And I highly recommend that you make sure that you view those additional documents, the NPR interview with Jimmy Carter, the Guinea Worm eradication strategies, and Dr. Campbell's Nobel lecture. So thank you very much.